Hello everyone. Thank you, Mike, for nice introduction. So welcome to our webinar. Today we will talk about using of the CIM Monolithic 96 well plates for process optimization of recombinant AV downstream process. Actually, I'm coming from Sartorius company, and our team is simplifying progress. And with using of this monolithic 96 well plates, we really simplify the progress of our process development. So first, so a little bit about our company. So we are named Sartorius Bioseparations. We are located in Slovenia. We are located in Slovenia in the town called Ajdovščina, here more on the west part of Slovenia. We are exclusive producer of convective interaction media, monolithic chromatographic columns. So we are working on this since 22 years, and now we are more than 200 employees. So what is our specialty? So we are focusing on the purification of large biomolecules. With large biomolecules, we think viruses such as AV. We will talk more about this today. Adenovirus, influenza virus, basically any virus that can be manufactured or it needs analytics, as well as other large biomolecules, nucleic acids, so plasmid DNA or mRNA. Also exosomes, VLPs, and basically the variety of large biomolecules is very large. Since 2020, we are merged with the Sartorius company. So this is why we are called Sartorius Bioseparations. And after this merge, we become Sartorius Center of Excellence in Gene Therapy. So in gene therapy, many large biomolecules are used. So we are working together here in offering solutions for complete downstream process development. So also services, solution for manufacturing. So these are our columns and scale-up systems, as well as analytical methods development that are used in gene therapy. So how the monoliths actually work? Why are they so unique? So here on the left side, we see a traditional approach with beads chromatography. So we see here beads with the pores that actually are often too small for large biomolecules to enter. And this causes very low capacity for the large biomolecules, as well as here in the beads chromatography, it is diffusive mass transport. With our novel monolithic approach, so with the monolithic columns, we can imagine something like sponge-like structures with the channels of large diameters. So these channels usually are, our typical diameters are 2 and 6 microns channels. And these channels have a lot of functional sites on all the surface where these large biomolecules are migrating in the solution. So we have very high accessibility of functional groups for the large biomolecules. So this causes very high capacity. Another very important thing is, of course, convective mass transport, what causes the flow independent resolution and also capacity, simple scale up, and it results in very fast process. So with these monolithic columns, we can see that they're really ideal for the purification of large molecules. They're constructed in this way. So our portfolio of products and services is very wide. So we have chromatography preparative columns. These are the columns that are used for the small scale, like 1 milliliter, up to large industrial manufacturing volumes, 40 liter. So these are, is very large portfolio of these preparative chromatography columns, and this Simultus line is scalable. So we can design the process on 1 milliliter column and then transfer it to the manufacturing up to 40 liter scale. They can be single use or multi-use. They are ready to use. We don't need to pack them in the housing. And for manufacturing, they are also CGMP compliant. So another product that we will talk more about it today are CIM Monolithic 96 well plates. This is the product that have 96 wells, and each well contains a small monolith that has the same material as large preparative chromatography system. So these plates are used for high throughput screening, design of experiments, and yeah, basically they can be used in the process development in a lot of different examples. So for these plates, we do not need chromatography system. We can use them with other techniques I will explain later, as well as their 96 well format enables that they are compatible with other standard liquid handling equipment. So this calls for the process automatization. Then another, let's say, side of the products is analytical. So we have analytical HPLC system, PATFIX, that is connected with a special software, and also is a triple detector system. I will also talk a bit about it later. And to this PATFIX system, we can attach our analytical columns, CMAC line, that these are small column volumes in the stainless steel housing that can be used in the HPLC systems. Since they are also monolith, they enable short analysis time compared to other HPLC columns, and we have specialty based on the application. So we offer CMAC PDNA column, AV, or Adeno column. So these are specialized for the application. So since we have this large variety of chromatography solutions, we also know how to use them. So we offer also the services. So we offer services for the process development, as well as for the analytical method development. Yeah, so CIM Monolithic 96 well plates, we will talk more about today. So here is the picture of them. They are very robust. So as I said, the same material as it used for the large chromatography column. They are reusable or single use. They have very high uniformity between the wells. So all the wells are very similar to each other. Since they are 96 well format, they can be used with other equipment that is formed for the 96 wells, and they can be operated using vacuum. They handle up to minus 0.06 megapascal, but working range 0015 up to minus 003 megapascal. If we use centrifuge, we recommend using the force of 500 Gs and maximum 1000 G for the operation. They're from polypropylene and polyethylene material. As I mentioned, these are chromatography resins inside, but the matrix inside, but actually we do not need the chromatography system for operation. Basically, we use the vacuum manifold, centrifuge, or positive pressure. And we operate them the same as the other chromatography for the chromatography column. So we first remove storing solution, then equilibrate them, we load sample, wash, elute, we can perform strip, and in case of reusability, then regeneration and storage. So today we will talk more about purification using monoliths of recombinant adeno-associated viruses, so shortly called AVs. So in our company, we developed the platform process for AV purification. And here the main challenge is how to separate AV and also polish it from the very starting material that is usually lyse harvest from the cells. And these lyse harvests have very high concentration of impurities from the host cells relative to the target product. So for this, we need the capture step. So here is the capture step, and then first the sample is prepared. So in this pre-capture step, we try to minimize also or at least lower the amount of impurity. Then SO3 elution from the capture step is diluted or buffer exchange. And then we have the polishing step for the separation of full and empty capsid. So for polishing step, we can either use Simultus QA, Prima S, or Prima T column. And then just the formulation is left. So today we will focus on the capture step. So this capture step is done on the pH 3.5 or higher. So usually we work in the acidic pH, so the AV is positively charged at that pH and binds to the cation exchange column. And in this capture step, majority of impurities are removed. So why we need to remove impurities? So first to enrich our product and also to achieve better polishing step that is following the capture. We have very high capacity. And as I said, this is a platform process, so it binds any serotype. But still, there are small changes between the serotypes, so we need to fine-tune the capture step with the buffers. And actually, this is what we did in our laboratory using the CIM Monolithic 96 well plates. We use them also to test the different chromatography conditions. So these are the DOE studies. So for DOE studies, they require a lot of different testing conditions. And with using the plates, we also lower the time needed to perform these experiments, and also we need smaller amount of sample. We also check different sample preparation technique prior to the SO3 column, for example, effect on the additives. So if we can achieve better recovery and so on. So in our example I will talk later, we had sample preparation, then we use the CM96 well plates for the screening of the capture step, and then these results we transfer to the Simultus SO3 column. So yeah, our experiment was designed in the way to define the binding conditions for AV9. So here we took the 4 pH that we test. So we test pH 3.5, 4, 4.5, and 5, and 4 different salt concentrations. So from 0 to 750 millimolar, as well as the presence of the poloxamer. All conditions were tested in triplicates. So we also we put all of these conditions in 196 well plate. So the presence of poloxamer, this was studied to see if it has an effect of the process recovery. We use this in the laboratory scale. So we had the classic vacuum pump and the manifold where we attached our 96 well plate. So we acidified sample to the target pH, so either 3.5 or 4.5 or 5. Then we load all the samples to the plate. Then we wash it with the buffers that have adjusted salt concentration, pH, and poloxamer. So this was the wash fraction. And then we elute with a buffer of the same composition, but 2 molar sodium chloride for the elution. So after we obtained the wash and elution fractions, we perform ELISA screening for the titer determination, SDS page to check qualitatively the impurities. We did the plate reader fluorescence, as well as HPLC analytics. So our results are showing. So this is first the fluorescent reader on the top, fluorescence reader in the elution fractions. So we were checking tryptophan fluorescence here. And we could see that we have basically two populations showing in the plate. So one is here showing in more dark yellow that has a higher fluorescence values. And then we have also the population with the lower fluorescence values more on the lower right bottom. So this indicated that maybe this means that higher fluorescence value means that AV is bound to the monolith, and in the lower fluorescence value, AV was not bound anymore in this condition. But we need to know here that also impurities are protein impurities, so they have also their own fluorescence signal. So most likely this positive fluorescence signal is combination of all of these. We were confirming our assumptions also with ELISA. So we took the positive sample and then the two positive sample for each pH and one negative sample. So, and here we can see that in the red are colored lower ELISA titer. So indeed, lower fluorescence was meaning the lower titer. And yeah, we got values for all these wells. We also perform SDS page. So in the SDS page, we can see that we got very clean fractions from almost all pH. So here is always the load sample and then the elution samples. And we can see mainly just elution fractions are showing VP3 bands from the AV. Only the pH 5 was showing impurities also in the, were co-eluting with the AV in the elution fraction. Then we were performing analysis of all these results. So we took the SDS pH purity densitometry, fluorescence values, and ELISA titer. And we did statistical analysis. And we can see that here a statistical analysis determined that the best buffer A for the CX column for this sample is pH 3.5, 500 millimolar sodium chloride in the presence of poloxamer. So these molecular biology techniques took us quite a long time to perform after the for all the elution fraction. So we thought if we can do this also in a bit faster way. So we took the HPLC PATFIX system and our in-house CXAV quantification method. So this method is very good because in the same time it measures the AV titer and also the impurities. We had triple detector, so we have fluorescence reader, UV, and light scattering, so-called MALS detector. I will show here for examples of 2 wells, but we analyze all 96 wells here on the CIM SO3 analytical column. And we can see that, for example, this is a positive well, very top left well A1, where we obtained the AV peak and also CIP peak. So we were cleaning in place peak. So CIP peak, we target to have minimized and maximized, of course, the AV peak for each well. So, and then on the right hand is the demonstration of one of the negative well. So here we see we have no AV peak and also very little of the CIP peak in this case. So we were integrating all of these values. So all the Mal signal, UV, and the fluorescence for AV as well as for the CIP peak. So each of these methods lasted 12 minutes in this case. First, we check if we got some correlation between our, let's say, HPLC data and ELISA titer. So we see here, we obtained a correlation between area of the light scattering in the AV peak and the ELISA titer, as well as the area of the fluorescence compared to the ELISA titer. So then we put just the HPLC results for the statistical analysis. So excluding all the results we obtain in the first way of analysis. So we just took the area of the AV peak and area of the CIP peak, and we got very similar results that we obtained also for the first way. So, but this was a lot faster than all the molecular biology techniques we were using in the first version of analysis. And moreover, this can be also automated. Later, very importantly, we transfer our results to the Simultus line. So this was just the plates were used just to optimize the buffer for the CX column. And then we perform also the preparative chromatography using Simultus line. So we see here a very nice chromatogram where we load the sample, wash the sample with a newly defined buffer. We perform elution. We see very nice sharp peak and no other peaks around the elution. So it looks really clean also from the chromatogram. And then we see also the CIP peak. So we were really optimizing these conditions with the plates. These fractions from the preparative chromatography we analyze with the picogreen for the total DNA content and also BCA test for the total protein. We see here that we reduce almost 100% of the proteins and the DNA in this fraction with still very high SO3 recovery that was here 87%. Our capacity here was 1E14 capsids per milliliter. So this is the dynamic binding capacity. And we really improved our in-house AV platform process with using of these monolithic plates. So results were also transferable to Simultus line. So the monolithic plates are actually very robust tool for this fast preliminary screening. And yeah, this is very important because with optimization of the capture step, we can really achieve higher process productivity and also the purity of the product at the end. So also less problem in the polishing step. But here I was just showing one example of monolithic 96 well plates that was used in our laboratory. Of course, there are endless ways of doing experiments with these plates. And yeah, we are very happy to use them in our process development department. So with this, I would like to thank you for your attention, and yeah, I'm happy to take questions.